Hi students, in this video we shall discuss about pyruvate oxidation from the lesson Respiration in Plants. At the end of the glycolysis, we have two pyruvate molecules that still contain lots of energy. Pyruvate oxidation is the next step in capturing the remaining energy in the form of ATP, although no ATP is made directly during pyruvate oxidation. So in eukaryotes, this step takes place in the matrix, the innermost compartment of mitochondria. In prokaryotes, it happens in the cytoplasm. Overall, pyruvate oxidation converts pyruvate, a 3-carbon molecule, into acetyl coenzyme, a 2-carbon molecule attached to coenzyme A, producing an NADH and releasing 1 carbon dioxide molecule in the process. What is the use of acetyl coenzyme A? Acetyl coenzyme A acts as a fuel for the citric acid cycle in the next stage of cellular respiration. Next, we shall discuss about steps of pyruvate oxidation, children. Before that, pyruvate is produced by glycolysis in the cytoplasm, but pyruvate oxidation takes place in the mitochondrial matrix in eukaryote. So before the chemical reactions can begin, pyruvate must enter the mitochondrion, crossing its inner membrane and arriving at the matrix. In the first step, a carboxylic group is used by the pyruvate and a molecule of carbon dioxide is released, leaving behind a 2-carbon molecule. Clear, children? A carboxylic group is used by the pyruvate molecule and a molecule of carbon dioxide is released, leaving behind 2-carbon molecule. In the second step, the 2-carbon molecule from the step 1 is oxidized and the electrons lost in the oxidation are picked up by NAD plus to form NADH. The third step is the oxidized 2-carbon molecule and acetyl group which is attached to coenzyme A. And what is a coenzyme A? It's an organic molecule derived from vitamin B5 to form acetyl coenzyme A. And sometimes this acetyl coenzyme A is called a carrier molecule, and its job here is to carry the acetyl group to the citric acid cycle. Citric acid cycle is nothing but Krebs cycle, children. The reactions are carried out by an enzyme called Pyruvate Dehydrogenase complex, which consists of 3 interconnected enzymes and includes over 60 subunits. Clear, children? So the reactions are carried out by an enzyme called Pyruvate Dehydrogenase complex. The Pyruvate Dehydrogenase complex is an important complex as it controls the amount of acetyl coenzyme fed into the citric acid cycle or Krebs cycle. So this pyruvate dehydrogenase complex is an important complex. It controls the amount of acetyl coenzyme A used in the citric acid cycle. We can summarize the pyruvate oxidation as follows, children. The 2 molecules of pyruvate are converted into 2 molecules of acetyl coenzyme. Clear, children? 2 molecules of pyruvate are converted into 2 molecules of acetyl coenzyme. And 2 carbons are released as carbon dioxide out of 6 originally present in the glucose. Clear, children? 2 carbons are released as carbon dioxide and 2 NADH high energy rich compounds are generated from 2 NAD plus. Clear, children? 2 NADH are generated from NAD plus. Finally, by the end of the pyruvate oxidation, 2 NADH molecules are generated. What is the use of acetyl coenzyme in this process, children? Acetyl coenzyme serves as a fuel for citric acid cycle in the next stage of cellular respiration, and the addition of coenzyme A helps to activate the acetyl group, preparing it to undergo the necessary reactions to enter the citric acid cycle. Clear, children? So acetyl coenzyme A helps to activate the acetyl group which enters into the citric acid cycle. I hope you have understood this process of pyruvate oxidation, children. We shall discuss about Krebs cycle in the next video. Thank you.